So next talk is from Suzanne Eaton from Dresden, and she will be telling us about, I guess, tissue mechanics, viscoelasticity, and more, and Drosophila for sure. Thank you,
Okay.
Suzanne.
Thanks, Thomas, and thanks for the invitation to such a really interesting meeting. So what I want to tell you about today are some of the molecular mechanisms that underlie how tissues deform in response to stresses and the importance of that in development. And just to start off by saying this has been a great collaboration with the group of Frank Ulischer over the years, and in fact, two of the people that have been involved with this work are actually here, so Raphaël Etourné and Guillaume Selbreux, so they can see how it's evolved since they were around. Okay, right, so what you're looking at here is the network of cell contacts labeled by E-cadherin GFP in the wing of a fly as it's shaping itself during the pupal stages of development. And what movies like this have told us, what really is highlighted by these movies is how dynamic the process of development is. So all of these morphogen signaling pathways that we know are important for controlling tissue shape, this is where they have to act in the end. They have to act by controlling the physical properties of cells and tissues. So we need to understand the patterns of cellular force generation during a process like this. We need to understand how this results in the emergence of tissue stresses, and then we need to understand how tissues respond to those stresses by changing their shape, so the sort of viscoelastic properties of tissues. And so the way that my lab thinks about this is we want to look at this at multiple scales. We want to know how these morphogen signaling pathways affect inside cells, molecular events that are responsible for their physical properties, how from this emerge patterns of cell dynamics, like divisions and rearrangements, etc., and then essentially how all of these dramatic morphogenetic changes emerge from these patterns of cell dynamics. So we take a multi-scale type of approach. Okay, so the process that I'm going to be telling you about today is this process of pupal wing remodeling, and this is something that was first described by Benoit Aigui when he was in my lab a while ago now. So what's happening at this time, let's see, is this purple region that I've colored in here, which is going to give rise to the wing hinge. That's like the little region in the shoulder. Okay, and this pink region is going to give rise to the wing blade. At the beginning, they're about the same size, but what happens is that this hinge undergoes a dramatic pattern contraction, and then this blade region elongates and in the proximal distal axis and narrows in the anterior posterior axis as a result of these anisotropic tissue flows that you can see here. And this is a really good example of a tissue deformation that's happening in response to stresses. And what we learned about this, and this is the work of Raphael and Guillaume and other people in the lab at the time. What we learned that happens at this time is that when the hinge contracts, that results in the emergence of proximal distal oriented tissue stresses that we can actually measure by this kind of laser ablation that you've already heard about. And that happens because the wing blade is connected to an overlying cuticle by an apical extracellular matrix protein called dumpy. So when the hinge contracts, now you get a counterforce and you get these stresses, and they build up when the hinge contracts. And now these are some laser ablations we've performed over time. This was done by Vinky Iyer, a postdoc in the lab. So the stresses build up when the hinge contracts, and then they gradually relax as the blade is changing its shape. And they pretty much completely relax by the end. So the wing blade is behaving like a fluid here. This is a viscous deformation. The stresses have relaxed by the end. Okay, so what are the cell behaviors that underlie this viscous deformation? What are the cells doing to allow the wing epithelium to behave as a fluid? And to approach this question, we do these we segment and track every single cell in these long-term time-lapse movies using this tissue analyzer that was built by Benoit, and also we analyze the data now in a computational framework called Tissue Miner that Raphael built when he was in the lab. And what this allows us to do is follow things like cell lineage, cell rearrangements, keep track of who's a neighbor with who, quantify properties like area and shape. So for example, here you're looking at this movie, but now we've colored in all the cells depending on how elongated they are. So red is more elongated, blue is less elongated. And you can see a lot of interesting changes in cell elongation happening at this time. So if we want to understand how much each of these different kinds of cell dynamics contribute to the change in shape of the wing, it's not enough to just, for example, count up cell divisions and how oriented they are, or count up cell rearrangements and which way they went. To really quantitatively decompose the shape change of the tissue into the underlying cellular contributions, you need to do a bit more than that. And this is a method that was devised by Matthias Merkel when he was a student with Frank. And I can explain this in more detail later if you're interested. This is what also Guillaume was talking to you about earlier today. Here we're able to decompose shape change in a region of interest of the tissue quantitatively into contributions from changes in cell elongation, cell neighbor exchange, cell division, extrusions, and this last term, which is a correlation term that I can also explain later if you're interested. And so we can either now look at patterns of shape change in the wings. So for example, here is one time point in that movie where each of these little blue lines represents just locally which direction the tissue was changing shape and how much it was changing shape. And then we can decompose each of the shape changes into the underlying cellular contributions and their patterns. But to look at really what's happening over time, it's much easier to average all of these contributions over the entire wing blade, and that's what you're looking at here. So what this graph represents is the cumulative change in tissue shape and the cumulative cellular contributions to it over this entire process. And now, if the change is positive, that means that it's contributing to elongation in this proximal distal direction, and if it's negative, it means it would tend to elongate the tissue in the orthogonal direction. So if you just look at this blue line, which represents the shape change of the whole, just the whole shape change of the wing, you can see it's getting longer in the proximal distal axis and narrower in this orthogonal axis. And here are all the different cellular contributions in different colors, and all of these curves add up to that blue line. Okay, and so if you just look at the end of the process, then you can see that the shape change of the wing was due to changes to neighbor exchanges, so red, changes in cell shape, this green curve, also in part due to oriented cell divisions, and this correlation term actually contributes somewhat. Okay, and what you also see is that the pattern is a little bit more complicated than it had to be. So for example, just note this kind of interesting complementary relationship between cell elongation and neighbor exchange. And so overall, what's happening during the process can be divided into two phases. In the first phase, cells are actually undergoing neighbor exchange in a direction that would tend to elongate the tissue vertically, okay? But the wing is not changing its shape vertically because it's constrained by a cuticle, and as a result, the cells are elongating orthogonal to that. And so that's what you can see here, and we know that cells are also elongating in response to the contraction of the hinge. Then now what happens during phase 2 is that the process reverses itself. The cell rearrangements turn around and now begin to lie along this horizontal axis, and that partially relaxes the shape of the cells. But by the end of the process, both neighbor exchange and cell shape changes have contributed to the shape change of the wing, and because the stresses in the wing have relaxed, we know that, for example, these cell shape changes are most likely a viscous deformation of the cells. Okay, they're not, if you cut them, they would not spring back. Okay, so we can actually look at which of these processes depend on the stresses that are caused by hinge contraction by interfering with these stresses and doing the same kind of analysis. So for example, if we take this mutant dumpy, where you now can't make these connections to the overlying cuticle, and when the hinge contracts, the whole blade just pulls away from the cuticle and you don't elongate it, now we can just analyze the cell dynamics in these mutants and see which of them have been disturbed by relaxing the stresses. And essentially what we find is that some component of the proximal distal cell elongation is disturbed by getting rid of the stresses, and also the realignment of cell rearrangement with this proximal distal axis. That also does not occur when you disturb the stresses. So these are two kinds of cell processes that would contribute to the fluid-like behavior of the wing, we think, in response to stress. Okay, so what I told you, strongly implied that the cell shape changes were really a viscous deformation. So how could we really study the viscoelastic properties of cell shape? Well, if you were a material scientist, you might take your material and subject it to some kind of oscillating stress, and then you would look to see how it deformed in phase or out of phase with the applied stress. So if you were a purely elastic material, and here's the shear stress, then you would deform just in phase with the stress, and if there were a viscous part, then that would shift the phase of the deformation with respect to the stress. So we can just exploit the natural buildup and relaxation of stresses in the wing epithelium to look at cell shape viscosity. And so what you're looking at here in purple is the stress. So this was determined looking at the initial recoil velocity after laser ablation, and this is over time as this process is occurring. And in green, you see in this same region that we cut the cell elongation at that time. And you can see that there's about an 8-hour delay between the peak of the stress and the peak of the cell elongation, suggesting there's a strong viscous component to the mechanical properties of these cells. Okay, so what kind of molecular mechanisms could allow cells to change their shape and neighbors in response to stress? So both of these things involve remodeling of the cell contacts, essentially. And so we highly suspected that maybe there could be some increased turnover of junctional components like E-cadherin. So what Venki did when he came to the lab was begin some FRAP analysis of E-cadherin GFP during this process. And so what he found was he would bleach individual cell boundaries, look at recovery, and depending on the time during this process that he did this, he could see very strongly different kinetics of recovery of E-cadherin GFP. And we can fit all of these curves very well to a double exponential, at least over the time scale of 8 to 10 minutes or so. There's one time scale of recovery that's on the order of minutes and another that's on the order of a few seconds. And if we wait for a couple of hours, we can get 90% of the recovery back. So there's a third pool that is relatively immobile that we don't access by this kind of experiment. Okay, so now if we look at these two different pools, the minute time scale recovery pool and the second time scale recovery pool, and compare them with the stresses over time, we can see that this minute time scale pool sort of increases and decreases in line with the buildup and relaxation of stress, suggesting, okay, maybe it's depending on the stress somehow. And so to look at that, one thing that Venki did was simply to measure E-cadherin recovery in these dumpy mutants where the stress is less. And indeed, what he sees is that this minute time scale pool of E-cadherin is decreasing, and the immobile pool is increasing when the stresses are reduced in a dumpy mutant. And another alternative way of doing that experiment is simply to do a FRAP experiment before and after a circular laser ablation. And then if he does this experiment, what he sees is that, as soon as he can measure it after he does the circular laser ablation, this minute time scale pool of E-cadherin is reduced and the immobile fraction increases. Okay, so that would suggest that the tissue stress enhances the minute time scale turnover of E-cadherin. So what are these different pools of E-cadherin anyway? So you could imagine two different sources of recovery for E-cadherin on this cell boundary. You could either get some diffusion from adjacent cell boundaries, perhaps, or you could get some delivery to that cell contact from an intracellular pool. And so to get an idea of which of these things were contributing, what Venki did was to bleach adjacent cell boundaries, and that should get rid of that source, or to also bleach the inside of the cell, and that should get rid of the vesicular pool. And what he found was that the minute timescale pool was very sensitive to bleaching inside the cell, while the second timescale pool was sensitive to bleaching adjacent cell boundaries. So we think that diffusion diffusion along the membrane is giving you the second timescale recovery pool, and some kind of vesicular delivery is important for the minute timescale pool. So you could imagine newly synthesized E-cadherin getting delivered by vesicles, or you could imagine internalized and recycling E-cadherin being delivered. And so we wondered maybe whether some of that indeed might come from endocytosis and recycling. So to look at that, what Venki did was to ask whether this fraction was affected by Shibiri temperature-sensitive mutation. So this is a temperature-sensitive mutation that blocks endocytosis. And so what you can see here at the permissive temperature of 25 degrees, both the wild type and the mutant have similar kinetics of E-cadherin recovery. But now if we raise the temperature to the restrictive temperature, number one, something interesting happens to wild type, which I don't have time to go into, but I find this really super interesting, is that when you raise the temperature in wild type, the mobile pool of E-cadherin increases. It's really interesting when you start to think about temperature acclimation in some way. Okay, but this is what happens to wild type. But essentially, in a Shibiri temperature-sensitive mutant, you have a lower mobile pool of E-cadherin, and that is specifically this minute timescale recovery pool. So, so we think that this minute timescale pool really is dependent on endocytosis and recycling. Okay, so just to summarize what I've told you so far, there's a fast recovering pool that diffuses in the membrane, a minute timescale pool that's coming from endocytosis and recycling, and a slower pool that is somehow protected in some way from endocytosis, and tissue stress changes the balance between these two pools. Okay, so how might that happen? So endocytosis of E-cadherin. Well, one thing we know regulates the endocytosis of E-cadherin is the binding of a protein called P120 to the cytoplasmic tail. And it binds at the same site that the endocytic machinery binds. So when P120 is bound to E-cadherin, it can't be endocytosed. And so we wondered whether perhaps mechanical stress in the tissue might affect the binding of P120 to the E-cadherin cytoplasmic tail. So what Venki did was to simply stain pupil wings for P120 at different times, at times when the mechanical stress was high, and then as mechanical stress relaxes. And what you can see is that P120 is much more cytoplasmic under conditions of high tissue stress, and it goes more to the cortex under conditions of low tissue stress. So that was interesting. Okay. And so we wondered then what would happen if you would take away P120 and now look at the stress dependence of E-cadherin turnover. So here, what Venki did was simply to look at the peak of tissue stresses, that's blue, and after stresses have relaxed, that's red, in wild type or in this P120 mutant. And you can see that this stress-dependent difference is gone, and that now E-cadherin is turning over fast, independent of whether the tissue stress is high or not. Okay, so what does that do to the viscoelastic properties of the tissue and of cells? So now let's go back to that method I showed you before, where we look at whether or not cell shape is deforming, in phase with the stresses or not. So here was wild type, and I showed you that there was an 8-hour delay between the peak of the stress and the peak of the cell elongation. But now if we look in a P120 mutant, now there's only a 2-hour delay between those peaks. So that would suggest that the P120 mutant tissue cells anyway are just less viscous. And to be a little bit more quantitative about that, we first asked whether our data would fit a Kelvin-Voigt model. So here we would say that, okay, tissue stresses would be a function of some kind of shear elastic modulus times the cell elongation plus some kind of cell shape viscosity multiplied by the rate of change of cell shape. And so we find indeed that the data do fit a Kelvin-Voigt model very well in both cases, and if we look at the viscoelastic relaxation time, it's about 4 times higher longer in wild type than it is in the P120 mutant. So they really do seem to be have a lower viscosity. How am I doing for time?
You're doing good.
Good. Good. I like to. Good is good. Okay.
Keep going.
Okay, keep going. All right. Okay, so I just showed you about cell shape. Now,
4 minutes and a half.
Until I should stop talking, or until I don't have any.
1.5 until you stop.
Okay, 1.5.
You have 4 minutes.
Okay, I have 4 minutes. Good. Okay. Right. Okay. Okay, so you might ask yourself also what would happen to the other kinds of cell dynamics in a P120 mutant. And so this is just the same kind of decomposition of tissue shape change into the underlying cellular contributions that I showed you before. But now we're just doing it in this one region where we've done the laser ablations. So we know we're looking at exactly the corresponding piece of tissue. That's why it looks a bit noisier. And essentially what you see in the P120 mutant compared to wild type is that everything is just compressed. It's just everything is happening faster. The buildup and relaxation of stresses looks the same, but everything is just happening faster. The cell rearrangements, the cell shape changes, it's all happening faster in a P120 mutant. So this, all of this has just been published recently, and so you can, but what I maybe will spend 1 or 2 minutes telling you about now is some more unpublished work about how planar cell polarity complexes also contribute to somehow the mechanical properties of this tissue. And we know much less about this, but I just can't resist telling you a little bit. Okay. So planar cell polarity, you probably know it best because this is a system that's important for orienting external structures like hairs and bristles. And that's one thing it does in the wing. If you get rid of this system, now you have wing hairs just pointing in all directions. But what you also have are changes in the shape of the wing. So this is just a prickle mutant. And if you look at the major-minor axis ratio in these wings, it's reduced, and there are also some problems with cross-vein placement. So there's a funny effect on wing shape as well. So what are these complexes? I'm going to focus today on the core planar cell polarity pathway. So this organizes junctional complexes that are polarized within cells and asymmetric across cells. And they can organize, self-organize polarity in ways that they link the polarity of adjacent cells, and then they can form global polarity fields in tissue. And we've studied how these global polarity patterns evolve over time during this process that I've just told you about. And what we know is that at the beginning of the process, these core PCP domains point towards the wing margin. And then as these flows happen, the core PCP domains reorient to point along the proximal distal axis. And here you can see this is just correlating the orientation and polarity of these complexes with the cell elongation. And you can see it happens precisely in concert with this cell elongation and relaxation. So we wondered if somehow these complexes could play a role. Also because we know that their reorientation depends on these tissue stresses, because if we cut the wing blade off of the hinge before this process happens, now we completely develop a weird global pattern of planar cell polarity. And so when a graduate student, Franz Kluber, was in the lab, he had actually done this kind of cell dynamics analysis in some PCP mutants. And it's remarkably normal, but what you see is at the end of the process, the cells keep relaxing their shapes way too much. And that means that even though the wing elongates pretty normally at the beginning, it kind of starts relaxing back the other way towards the end. And you also have an effect on these oriented cell divisions. So what is it actually doing to the mechanical properties of the cells? Well, what Romina Piscitello has done, she's also a graduate student in the lab, is she's done this same kind of laser ablation plus cell elongation analysis on all of these different planar cell polarity mutants and compared it to wild type. And this really surprised us. This was not what I was expecting to see. So here are the stresses in wild type, this turquoise line building up and relaxing over time. And now look at all of these PCP mutants. They just don't build up the stresses properly. And then they come down a little bit, and then they start to go up again. But despite that, the cells are elongating, at the beginning perfectly normally. So, and the delay is, so it's not like in a P120 mutant, there's still a delay. But what I think this must mean, and we're just in the middle of analyzing all of this, but what I think this must mean is that these mutant cells have different elastic constants or something. They elongate a normal amount, but in response to much less stress, interestingly. So we're just in the middle of trying to figure out how these things are influencing myosin organization. But I'll leave it right there and just leave you with my conclusions, which is that stress-dependent cell shape changes and rearrangements allow the wing epithelium to behave as a fluid, and that this is depending on the stress-dependent association of release of P120 from the cortex and into the cytoplasm, which affects E-cadherin turnover, and that the core PCP pathway has an interesting function in all of this, but we don't know what it is. So with that.